Thank you, Christian, for the introduction. Yes, thanks for the invite also to be able to present my work here. So I'm going to talk to you about the love life of a single cell, or evolutionary constraints for self-compatibility. And indeed, I'll be mostly talking about this yeast that you see here in the bottom left on the screen. So let's start immediately with, let's get my focus on the PowerPoint. What is self-compatibility? What do I mean with that? So when we're talking about self-compatibility, sexual self-compatibility, and it's all about if a cell is able to mate with itself, as the word says already. But as a consequence of this, self-compatibility will also mean that the cell is not able to distinguish others from itself, and it is able to mate with all other individuals. And because all individuals, we assume that they all have the same genetic background, they will also not be able to distinguish self from others. So everybody is able to mate with everybody. So mating between like will be possible here. If we just look at this focal individual here, we can try to figure out what the population level compatibility is. So if there is no, or if there are no different types, this will mean that all individuals are completely compatible with all other individuals. So 100% compatibility at the population level. If a cell is able to distinguish self from non-self, and it's only able to mate with other types, then it will only be able to mate with those individuals that are of different type. And as soon as we have two different compatibility types, the population level compatibility will drop by half, because this focal individual is only compatible with half of the population. If we increase to a third mating type, we will have 66% compatibility. This will, of course, increase with the more different types that we add to a population. And if we have an infinite number of compatibility types, we approach 100% compatibility again. So as you can see, we have here a drop from, if there will be no types, to 50% compatibility if there will be two types. And then the increase there will be an increase again of compatibility. So what we can say from this is that sort of the worst number of compatible types that are out there is 2. Nevertheless, we see that wherever we look, we have almost always 2 different types. In the animal kingdom, plants, brown algae, fungi, wherever you look, there will be 2 different, almost always 2 different types. And very few 0, and even fewer times there will be more than 2 different types. I'll be focusing on those groups where there will be, where there's no differences between sexes, so isogamous lineages. And these lineages still can have different types, and in this case, we will call these mating types. So definition, it's good to add a definition. In isogamous lineages, alternative haploid genotypes that determine compatibility in the sexual cycle. I added the word haploid here because the state of the gamete, their compatibility is defined at the haploid state. And I'll be mostly talking about fungi. A little bit more of terminology. I will be talking a lot about homothallism and heterothallism. Homothallism means that there's only one mating type, which is the same as there are 0 mating types, because everybody is compatible with everybody. So homo, the same, are compatible. Heterothallism is anything where there are more than 0 mating types, so 2 up to many, and this is called heterothallism. So you need to have 2 different hetero individuals in order for there to be compatible mating. The main questions that my group works on are questions, what select 4 different types? So how do we make this transition from what we assume was the ancestral state, 0 types, to 2 types? Second question we're trying to understand is, if we have two different types, right? This is sort of the worst state that you can be in. What would maintain these two different types? Why do we not switch back to no types? Or why do we not go to more than two types? And then the third question is, okay, there are circumstances where this does happen. Under which circumstances do we get fewer or more different types? The final question, trying to answer using this mushroom Schizophilium communi. This is still quite recently started research. Schizophilium communi is quite a cool little mushroom. It has about 23,000 different types, or maybe effectively something around 100, depends who you ask. So we're trying to understand, how did this mushroom evolve so many different types? I'll not talk about this today. Today I'm going to talk about the transition from 0 to 2 and back to 0 again. And for this we use the other yeast, Schizosaccharomyces pombe, Fission yeast. I'll tell you a little bit more about Fission yeast later. So the talk today has two main parts. The first part is going to be about, how does self-compatibility arise? So how do we get from a state where we have heterothallism to two different types? How do we drop back to 0 types? I'm going to talk a little bit about some lab experiments we've been doing and some measurements from a natural system. And so this is the love life of a single cell part of the title. And then after that, we're going to continue with talking about constraints for evolution. So what maintains cells at two different mating types. And from that, we're going to try to understand maybe why originally mating types have evolved in the first place. So let's start with talking a little bit about fission yeast. So as I already said, fission yeast, it's not budding yeast, but it's the other yeast. So it's not used as much for evolutionary research, especially as Saccharomyces cerevisiae, which is here in this group. So we're working with Schizosaccharomyces pombe. And it's important to note that these two are quite distantly related. So it's somewhere, it's more than 300 million years ago that these diverged from each other. So a lot of things that you might know that apply to budding yeast do not apply to fission yeast. But then again, a lot of things do apply again. So we can learn a lot from these two independent systems. So the fission yeast life cycle, the reason why we work with fission yeast and not with budding yeast is because fission yeast is predominantly haploid. So it's we have a haploid cell. The cell can grow by, well, as the name says it, fission. So this happens especially under rich environments. We end up with two cells, and these cells can keep growing till at some moment they run out of nutrients. We can also induce mating, similar to what would happen if the cells run out of nutrients. So we can move the cells, bring them in a low nitrogen environment. The two haploid cells will be induced to mate. We end up with a diploid zygote. The zygote will go into meiosis, and we end up with haploid spores. And these spores have a resting phase, so they will survive for a prolonged period of time. And when the environment gets better again, we can induce them to germinate and to continue again with their asexual cycle, or to continue with mating. And of course, we're interested to work with mating types, and this fungus does have mating types. We have two different types. In this species, they're called plus and minus. A minus cell is only able to mate with a plus cell, and vice versa. They're defined by a single locus, which has only two alleles.
Sorry, is it very similar to Cerevisiae, this system, or is it also mating type locus, the same thing, or is it done differently?
No, it's very similar. The genes are slightly different in them, but there are two alleles, single locus. And also the mating type switching that I'll talk about later is also very similar. They evolved independently, but many of the things are very similar here, yeah. for all ascomycete fungi, it's very similar. Evolution of homothallism. So to understand why homothallism might have evolved, we can think about the two main things. So for my first slide, right, so there's two things that are happening. If there is homothallism, we have universal compatibility. All cells are able to mate with everybody, but also the cells are able to mate with themselves. And these are two different evolutionary forces that might have led to evolution of homothallism. They're not mutually exclusive, but let's have a very quick look about at what might have been the driving force for what might be the main driving force for evolution of homothallism. Let's start with universal compatibility. So one of the arguments is that there's an increased chance to find a compatible mate. This is definitely true. I think it's not a very strong argument, and I will show you why. So if you would just think about if there is random mating happening, cells they move into a certain environment, and they just have to wait to find another partner. If we assume that there's an equal frequency of the two different alleles, as soon as we end up with a few different individuals per patch, we will see, what you can see here is the frequency of the number of compatible mates that are present in a certain patch for the total number of individuals in a patch. And as soon as we end up with quite a few number of individuals, and 30 is a lot for a microorganism, we're very close to 50-50 ratio, and most of the cells should be able to find a mate close by. So I think that this is true, but only under very low densities. This will play a role. So when there's a few number of individuals per patch. And one of the problems with fission yeast as well as with budding yeast, very often we don't really know what happens out there in nature. So maybe I'm completely wrong, and maybe the lower range is actually the total number of individuals in a patch. But I think it might be higher. So that's one of the arguments for universal compatibility. The second one would be that increased outcrossing might occur if you're compatible with everybody. It's also possible that you are more likely to mate with someone who is not you. Well, actually, if you do some simulations, so we did some structured cellular automatons, and if you look at this under all circumstances, when you increase the amount of homothallism, so switching efficiency, we'll see a little bit later why this isn't a measure of homothallism, so it's sort of a proxy for homothallism. So the more homothallic you are, actually the less outcrossing there is. This is both relative as well as absolute. In absolute numbers the case. And this is mostly because as soon as a cell has mated with itself, it's no longer available to outcross with another individual. And a heterothallic strain always, by definition, has to outcross. So that's why we are here at 1. So homothallism actually reduces the amount of outcrossing. It will not increase outcrossing. And also, if you look at this experimentally, we took some different yeast strains that have different amounts of homothallism. And if you try to outcross them, you can see that with an increased amount of homothallism, so in this case, homothallism is on this side of the range, we do not see that there's an increase in the amount of outcrossing. So these are a bit preliminary numbers. We're still trying to up these numbers. So far, it looks like there is no increase during outcrossing. So I do not think that evolution of homothallism is driven by universal compatibility. I would say it's most likely a result of the increased self-compatibility. So the ability to have intra-haploid mating. So let's dig a little bit into that. So self-compatibility, it might facilitate local adaptation. If you can mate with yourself and you are locally adapted, you don't break up beneficial gene combinations. And so this makes complete sense, I think. I've been trying to look a little bit if I could find some data for this. I cannot find it. If anybody has some good references, please let me know. So some research has been done in plants, but there's no clear signal of this as far as I have seen. We have done, this is a good moment to quickly mention another big project that I'm involved in. So we are running in the lab this together with Sergio Tuso and Jochen Wolf, who presented in this same series, what I understood from Christian. So you might notice system already. We have a big yeast experiment in which we have differential antagonistic selection for an ecological trait and different amounts of gene flow between the two divergently selected populations or subpopulations. And what we've seen in all of these populations is that there is evolution of self-compatibility. We have also seen that there is local adaptation. However, we have no clue in how these two are related to each other. It could be that they're completely independent from each other. We have at the moment a bachelor student who's trying a little experiment to see if there might be anything going on there. If you're interested about the experiment, was published last year, first paper on this. Okay, so second argument, and now we get into the love life of the single cell. Main argument, which I think is a reason why homothallism evolves, is that it helps with overcoming this loneliness. So this is the lonely spore hypothesis. It's sort of a microbiological version of Baker's law, which states that under low densities, organisms should lose self-incompatibility so that they are assured of reproductive output. You can imagine if you have a single haploid cell, let's say it's a plus cell. If this cell divides asexually, we now have two cells, but these two cells are now not able to mate with each other. If you would have an homotallic strain, the homotallic strain is able to mate with everybody, so also with a direct descendant of itself. So this is just the case if we would have a single individual, but also if we have multiple individuals in a certain locality, this will still be of importance. Homothallism will still be beneficial. Most of the microorganisms I'm talking about are sessile, so they can't move around. They can disperse by just growing. So they will form patches, and at a certain moment, they will no longer be able to grow further because they run out of space or they run out of resources or both. And at this moment, all the cells that will not mate, they will not be able to produce these long-lasting spores, and they will have zero fitness. But all of the individuals that are able to mate will produce offspring. So in this case, if we look at these plus and minus cells, only the cells that are located next to another individual of the opposite mating type will be able to mate. Whereas in homotallic individuals, all of the individuals will be able to mate throughout the whole patch. So this is something that we tested in the lab. So we have here 3 different strains. 2 of them are fluorescently labeled. The plus strain is fluorescently labeled and the minus strain is fluorescently labeled. And we have a homotallic strain, which is not labeled. So therefore, in this image, it's completely black. So we seeded these out at a low density. They were able to grow, and they will then able to form these patches. And then afterwards, we visualized where they had mated. So we can stain these plates. And whenever the mating has occurred, we get dark, to the spores color dark, whereas those cells that have not mated, they remain light. And as you can see, exactly as the model would suggest, where 2 heterotallic strains meet each other, we get a border, which is where the spores are being formed. But as soon as we have a homotallic patch, so a completely dark patch on the left side, we also have a completely dark patch on the right side, because that's where all the spores are, all the cells are able to find a mate and produce spores. To quantify this, we did some competitions between heterotallic and homotallic strains, and we did this under different dilutions. We're interested to see if there's also a density effect, because you can imagine if you have a lower density, these patches will become larger. If you have a higher density, the patches will be closer together and they will be smaller. And we did competitions. If we are at 0, it means that there's no difference between heterotallics and homotallics. Above 0, we have beneficial effect for the homothallic strain. And if it's below 0, the heterothallic would be performing better. As you can see, under all circumstances, and also when we change the ratios a little bit, we see that the heterothallics perform way worse. The homothallics completely outcompete them. So this is a selection coefficient from a single generation, and it goes up to almost 80%. So it's a huge fitness effect of being homothallic, and especially under these low densities. Additionally, we did some experimental evolution. So we started off with 2 heterothallic strains, and we let them go through 25 cycles of growth and mating. So we grow the cells asexually, then we let them mate. We kill off all the cells that have not been able to mate, and only the sexual spores are able to continue through the next generation. We germinate them again, grow them, etc. And then we checked afterwards how many of these strains contained homothallic strains for homothallic cells. And we saw that in 9 out of the 20 lines, homothallism had evolved. And in some of them, they even had gone to fixation. So again, we can say that there's clearly a very strong benefit of being homothallic in such a structured environment. So to summarize this, it's beneficial to be homothallic, especially under low densities. It helps because it assures that you will get a mate. And you can have increased mating efficiency by performing haploid selfing. So it's mostly about what happens within these patches, more than that it's about being compatible with all of the others. So that was the first part about why we would have transitions towards homothallism. These are the laboratory experiments. We also more recently did some analysis of natural variation. And for this, I want to talk a little bit more about the mechanisms that are underlying mating types in Fission yeast, and also a little bit more about mating type switching. So most of the mating type specific proteins, those are the ones that make if you're a plus or a minus cells. And so there's multiple genes that are defining your identity, but these are all under regulation of this single locus. So if you have at this locus a plus allele, then you will only express the plus proteins or the plus genes. And if you have a minus allele, you will only express the genes that are responsible for the minus specific proteins. Most of the genes that are involved in that make a cell a minus or a plus cell, that are mating type specific, are involved in communication between the cells. And in Fission yeast, it's what it looks like. All of the genes are involved in pheromones that are signaling where a cell is, and the receptors that are perceiving these pheromones. And there's some helper proteins also involved. So if you are a minus strain, you are producing genes that signal that you are a minus strain, and plus vice versa. But everybody carries all of the genes that are expressed in both of the mating types. This is relevant to know because Fission yeast, homothallism in Fission yeast is a bit special. It happens because of mating type switching. So if we have here 2 separate cells, we have a plus cell and a minus cell, and as already mentioned, the plus cell is a plus cell because at the mating type locus, it has the plus genes. And at the mating type locus, the minus cells have the M genes. However, next to this active mating type locus, there are 2 silent cassettes. And these cassettes are encoding information for both the plus as well as for the minus mating type. So what happens during a mitotic cell division, let's say we have a plus strain. Just at the active site, there is a small epigenetic mark, probably a single strand break, maybe a single strand break. And this break needs to be repaired. And in order for this repair to occur during the S phase, one of the two cassettes is used as a template to perform this repair. So if the cell was a plus strain, it uses the minus as a template, and if it was a minus strain, it uses a plus as the template, mostly. So the result is that we now end up with two cells. These are two daughter cells. They're genetically identical except for the information at the active mating type locus, and these two cells are now able to mate with each other. A lot of research has been done on this specific mating type switching phenomenon, and we know that switching is very efficient. It happens in the right direction, so plus becomes a minus and minus becomes a plus. Switching is very symmetrical, so there's as many pluses changing to minuses and vice versa. Everything is very efficient, mating is efficient, and we know that heterothallism can evolve, or switching can be lost, so we can end up with heterothallism again. So what I showed you before were switching strains and heterothallic strains. So this is all very nice, but we also know that all of this research has been done on a single lab strain. So everybody in the Fission yeast community is limiting themselves to a single strain, which is really nice. So all research is very comparable, but it also means that we might miss a lot of information. And people that have been looking at different strains have shown that there is quite some, potentially some variation here. So what we decided to do is to have a look at a large collection of natural isolates. So there's quite some paired and Illumina data available. We decided to look into this. We also had some of our own work in which we used short reads as well as long read data. And we use this to verify if what we did here is correct. And what we're interested to understand is how often do we see a plus at MUT1 or a minus allele at MUT1? So what we did is we just looked at this region right before the active mating type locus. We found all the reads that map in this region. This is a single copy region. A lot of the other regions are not. That's why we had to come up with a little bit of an elaborate method to test this. We just look at which reads are mapping in this region. We look at the pair and we ask ourselves, the pair, is it mapping to a plus, to a minus, or maybe to nothing? And then we just count how many of the reads are mapping to a plus or to a minus. As I said, we verified this using some PacBio long reads data, and the method seems to be good. And what we observed is that there is quite some variation in the total number of pluses and minus reads. So we see that some of them are completely minus. And if we look in the lab, we can also see that these strains are indeed heterothallic. So these are minus strains. We have some strains that are completely plus. And again, if we look in the lab, we see that these are mating as plus strains and not as minuses. But then we have a lot of individuals that are somewhere in between. And of these, only about 1/3 are actually not significantly deviating from a 50-50. There's quite a lot of them that have a bias towards the minus, but there's also some that have a bias towards plus. There's more plus individuals there. And of course, as expected, our lab strains fall smack in the middle. So it still completely follows all of the knowledge that we had about it. So then we can have a look at what does this mean. So we can sort of say that everything which is completely one type or the other is completely heterothallic. Everything which is intermediate has an intermediate amount of homothallicness or heterothallicness, whatever you want to call it. So the more biased you are, the more likely it is that you will not be able to find a mate of the opposite type. And we can see that if we just look at the mating efficiency, so mix a lot of these individual cells together, how many of the cells have mated. If there is very low switching, so this on the left side is heterothallic. These are completely heterothallic strains, and there's actually quite a few in this little dot there. Mating is very low, but when we have more homothallic strains, we can see that there's quite some increase in mating efficiency. We do see that there's quite a large number of strains that are very poorly mating, also even though they are asymmetric. Even though they are homothallic, and it's not really clear why this is the case. It seems to be many other factors as well that are involved here. Maybe they can't germinate well. Maybe they're able to, they're not mating very efficiently for other reasons. So just to summarize this first part about the love life of a single cell. So I've shown you in experimental evolution, as well as in this part on these natural strains, that haploid selfing is probably the main reason why self-compatibility might have arisen. It really increases the ability for you to mate, both if you are heterothallic versus homothallic, but also if you have different degrees of homothallism. The more homothallic you are, the more likely it is that you'll be able to find a mate. Okay, so now it's time to start talking about the second half of the title, which is constraints for the evolution of self-compatibility. And we're immediately going to add why this might, so the inverse is true of why we might have evolved to this situation in the first place. So we're going to quickly try to add that as well. I need to check my time. Let's see how it goes. So there are different ways in which homothallism can occur. I've talked already to you about the bottom one, mating type switching, but there are two other common terms for common mechanisms by which homothallism can evolve. And one is when there's single mating type mating. So cells that are of the same mating type are able to fuse with each other, even though there are also minor strains around where they might have a higher efficiency of mating with. Then there's a second part, which I call true homothallism. This is the type that fits best with the first described homothallic strains. And in this case, we have haploid cells, but these haploid cells have both of the mating type loci, mating type alleles present at different loci in the same haploid genome. There's still, in both of these homothallic types, we still have fusion is still required. So it's not that they are able to go through a life cycle as a haploid individual. There still needs to be mating happening, and we need to end up in a diploid phase. We think that mating type switching is an adaptation to overcome some of the problems that true homothallic strains have. So probably mating type switching is a way to not have to have both of these mating types together, but still being able to, from a single cell, be able to end up with two opposite mating types. We can get some fusion happening there. So let's first quickly have a look at this, what the consequences are of mating type switching. So we did some measurements just to see if mating type switching might have some costs. We know that it improves fitness during the sexual cycle, but during asexual reproduction, we again competed homothallics with heterothallics, and now we can see that under all circumstances, if we just have asexual reproduction happening, the homothallic strains perform much less good. So they have a reduced fitness relative to the homothallic strain, to the heterothallic strains. Sorry, and this, you can imagine that if you have to go through such a complex system, where you need to change part of your genome every mitotic cell division, this might incur some costs. However, we think that is actually, as I just said, it's an improvement on what happens in true homothallism. So there must be even more things complicated in a true homothallic strain. So let's have a look at what's happening there. First, we can think about what happens in these same mating type mating strains. So what happens here is we get problems with autocrine signaling. I'll try to explain it a little bit more. As I mentioned, we have plus and minus strains. The identity of the strains is defined by the mating type locus, but what happens at the what happens downstream of this is that there's interaction between the two cells that want to mate. That's what being regulated by the mating type locus. If you have a cell that's able to respond to its own cells, it's no longer able to distinguish self from non-self. It needs to respond in some way to its own pheromones. And if we make a simple schematic of this, we have here 3 cells that are just lying there on a little piece of agar or wherever they grow in nature. They will secrete pheromones into the environment, and you can imagine that where they are lying, that's where the gradient is, that's where the frequency, the concentration, so this should be concentration on the y-axis, so the concentration is highest. For these cells to figure out where another cell is, it would be nice if they could follow the gradient towards the highest concentration around them, but because it's highest because where they are themselves, they're not able to do this. If we would have a system where we would have 2 separate pheromones, they could just ignore their own pheromone they produce and only respond to the one which is of opposite type. They can just follow the gradient, and where the concentration is highest, that's where they should find a compatible mate. So this is one of my 2 favorite hypotheses of why mating types might have evolved in the first place. The gamete signaling hypothesis posed by Rolf Hoekstra and more recently got out of the dust by Zina Hadjivasiliou and Pomjankowski. And so they did a lot of modeling on this, and their argument is that it's very well possible that this could have led to separate mating types. So if we assume that originally there was a single mating type with 1 pheromone and 1 receptor, these 2 are not able to distinguish self from non-self, and it would have helped if at some moment they might have evolved different pheromone and different receptors. And there's 2 obvious pathways how this might have occurred. One could be that the pheromone and the receptor slowly have evolved through intermediate steps. This, of course, would reduce the efficiency of recognition between ligand, pheromone, and the receptor. But it could be a way in which this might have evolved. Alternatively, we can imagine that maybe already before the state that I drew in the top left, there is 1 cell that signals and the other cell that perceives. But this already assumes some form of asymmetry, and this might have then later on evolved, resulted into the system that we have here at the bottom. So we're at the moment trying to do some tests on this. So we have our fission yeast. There is quite a few different genes, but we know exactly which genes are involved in this mating type system. So we removed all of these genes and we've put them back in specific ways. So we have design strains which only produce their original, so a minus cell produce a minus pheromone, and their original minus receptor, and vice versa. We also constructed the inverted form, and if we mate these with each other, they're able to perform perfectly healthy and happy zygotes. We then went on to try to create symmetrical strains, so similar to the ones that I showed you at the top before. Again, it's possible to generate these and also to let these mate with each other. If we have incompatible strains, mating is not possible at all, exactly as we would expect. And if there is asymmetrical half compatibility, at the moment we've had some inconsistent results. So I'll leave a question mark here. It's not really clear. So sometimes there is some mating happening, but we need to verify this again. But at least what we have now is we have some strains which are what I think are these original strains. Original as in how evolution might have started. So what we're now doing is we're performing experimental evolution, trying to see if mating is increased. So mating of these ones is fairly poor if you compare it to the wild type strains. but we do see mating happening. And we've now done this for, I think, 12 generations. Students have been looking at this, and it seems that we do get an increased number of spores being produced. So that's promising. And hopefully we will be able to get some more information about which pathway we follow, if we go in one direction or the other, or probably it's evolution. So there must be pathways that we've never thought of. We might not end up here at all, of course. So that's how experimental evolution works. So that was the first main hypothesis and for the evolution of mating types. The second one results in, and this is a problem for these cells. This is both for truly homothallic strains as well as for the same mating type mating. But there are more problems if you put no or multiple mating types in a single cell. And this is because we will end up with some problems with cell regulation and sporulation. So let me explain that here. So as I mentioned before, a lot of the proteins that are under, or genes that are under regulation of the mating type are involved during the haploid phase in order for cells to find each other. But after fusion, the mating type still plays some role. So genes located at the mating type locus, at least in all fungi, they form heterodimers, and these heterodimers have a transcription factor function. They regulate, again, other genes that are only relevant during the diploid phase. And the argument from Haig, and later further developed by Nicolas Perrin, is that it's this the developmental switch that might have led in the first place to the evolution of mating types. So there are certain things that you definitely do not want to do if you are diploid, and there are things that you definitely do want to do if you're diploid. So you can imagine if you're a haploid, you want to produce all of the pheromones and receptors. You want to make sure that you have a nice mitotic cell division you can very rapidly produce. You want to mate with another cell in order to produce spores. But if you're a diploid, you want to avoid doing these things. As a diploid, you want to go into meiosis and you want to produce spores. In fission yeast, that's the main thing you want to do. Other organisms might, for example, switch from a single-celled life phase to a multicellular life phase. But what you do not want to do as a diploid is to continue with conjugations. But we would end up with a triploid cell, and if you then go into meiosis, you end up with a lot of aneuploid offspring. Haploid cells also do not want to do things that are good for diploids, so they definitely do not want to go into meiosis, because as soon as that happens, you end up with a lot of unhealthy aneuploid spores again. So the main idea of the developmental switch hypothesis is that you want to know you can use this one locus and these two alleles to see if you're a haploid. Right? So if you have only the plus or only the minus, you know you must be a haploid. As soon as you have both of these, you must be a diploid, because these two alleles are located at the same locus. So only if you are a diploid, you can be heterozygous at this locus. But if you are a haploid cell, a true homothallic, all of a sudden you have both of these alleles present in the same genome, and we end up with upper-green signaling. There might be some leakage of these pheromones that are still being produced. Cells might respond to their own pheromones again. There might be haploid sporulation. There's this very strong reduced growth. There's very low mating efficiency. Cells that, if you bring them onto a low nitrogen environment, they think they have already mated, so they will stop growing. They will no longer try to find a mate. So there's many problems here. And if you generate these strains artificially, this is exactly what you get. So we are working at the moment with some of these artificially generated true homothallics, just putting both of these alleles in the same haploid genome, and we get a lot of haploid sporulation, very strongly reduced growth, very low mating efficiencies. But what we do get in all of the strains where we introduce both of these in there, the strains are now able to reproduce by themselves. So some of the spores that are being formed are viable. So a cell, if you have a heterotelic cell and you let it grow by itself, it's never able to produce viable offspring. Whereas if you have one of these very sick, artificially generated homothallics, they are very sick, but they will be able to produce at least a few offspring. So it's very well possible that this is the first step in a transition towards homothallism. So to summarize this part, so evolution of homothallism is constrained by these two main functions that the mating types are responsible for. So the pheromone receptor system, which is in charge of mate finding and to induce conjugation between the two cells. And the two mating tableaus are also involved in the developmental switch, that the cells know that they should avoid multiple fertilizations, they should avoid haplosporulation, and they should be in the right cell cycle for either haploid or diploid phase. So that was the second part. So I'm going to give a very short summary of everything I've told you today. So first question that we asked was, under which circumstances might self-compatibility evolve? And this will be, at least that's what our experiments show, most likely when compatible mates are not available and you want to be able to reproduce, still have sex reproduction. And this is especially of importance under low density, because under those circumstances, because of the patch formation, you need to mate within, you want to mate within the patch, so you want to be able to perform haploid selfing. So what selects for different types? So at the ultimate level, it's most likely a mate finding that might have led to evolution of separate mating types, or possibly, and it's not mutually exclusive, there might be a role for ploidy assessment. So knowing when you have mated and knowing when you should stop mating. So these are the evolutionary questions. But then what maintains the different types? It's most likely proximate reasons. So there are constraints. And again, you want to be able to find a mate and you want to know if you are, you have mated already, yes or no. So we end up with the same answers. And that's all I had to tell you. There's some, of course, a lot of people that need to be acknowledged. People in the lab at the moment, my PhD student, Christina and Michaela, our TA, who have done helped out a lot with all these projects. And then there is a whole bunch of students that are specifically involved in this research. I specifically have to mention Rasha Schreim and Sebastian Schwartz, who have done a lot of the bioinformatics works and some of the lab work that I presented here. And then collaborators over the years, some funding. That was all I had to tell. A bit late, but I think it's time for questions, I hope.